Background information on macromolecules. This consists of carbohydrates, proteins, lipids, and nucleic acids. First up is carbohydrates, which are made of carbon, hydrogen, and oxygen and have three classifications of monosaccharides, disaccharides, and polysaccharides. Mono means one, di means two, and poly means three or more chains linked together by glycosidic bonds. These are the structures of the basic building blocks. Monosaccharides are the most basic. For example, glucose is a monosaccharide. Then disaccharides are two monosaccharides put together, for example, lactose. And polysaccharides are long chains of monosaccharides, for example, glycogen. These carbohydrates are fast and quick energy that is short-term for the body, and they tag proteins that allow materials to attach and help cellular communication. The next macromolecule is proteins, which are a little bit more complex and made of polypeptides. There are three classifications of proteins, which are fibrous, globular, and membrane. They're considered structural support, building blocks, or enzymes. Fibrous proteins are more narrow and lengthy and allow for more durability. Globular proteins are generally more denser and are weaker to temperature and pH changes. And membrane proteins have two types, which are integral and peripheral. Integral are pretty permanent and peripheral are temporary. Next is lipids, which are used to store energy. Fats and oils are typically triglycerides, which are made of fatty acids and hydrocarbon chain, for example, peanut oil. Phospholipids are polar and they make up the cell membrane. They also have hydrophilic heads and two hydrophobic tails. Sterolipids maintain the cell's fluidity and have an alcohol group that can help store energy, provide stability and protection, for example, cholesterol. And finally is nucleic acids. Nucleic acids are made of nucleotides. There are three subunits: nitrogenous bases, 5-carbon sugar, and a phosphate group, which make up the structure. There are two types of nucleic acids, DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. There are two types of nitrogenous bases: purines, which are a one-ring structure, and pyrimidines, which are a two-ring structure. Adenine and guanine are purine bases. Thymine and cytosine are pyrimidine bases. Guanine pairs with cytosine, and adenine pairs with thymine. For our experiment, our research question was, how does the temperature affect enzyme activity? The hypothesis was that if the temperature increased, then the enzyme productivity would too. The control was the room temperature vial. The control variables were the type of equipment, time of day, time intervals, quantity per sample, and the type of venom. The independent variable was the water temperature, and the dependent variable was the reaction rate. The conclusion was that our hypothesis was proved correct, that the enzyme activity increased more in the hot water bath than room temperature bath or cold water bath.
Gram-positive versus gram-negative bacteria. Gram-positive bacteria are made up of membrane proteins, the cell membrane, a thick layer of peptidoglycan, and lipoteichoic acid. Gram-negative bacteria are made up of membrane proteins, a cell membrane, a smaller layer of peptidoglycan, and an extra outer membrane. The outer membrane contains lipid proteins, porins, and polysaccharides. The gram staining procedure begins by adding a crystal violet dye. This dye then attaches to the cell membrane. Iodine is then added to secure the crystal violet dye in place. However, because of the outer membrane present in gram-negative bacteria, the iodine is unable to pass through and secure the crystal violet to the membrane. The decolorizer, or ethanol, is then passed through to wash away any unsecured crystal violet. Finally, safranin is added to indicate gram-negative bacteria. This shows a gram-positive bacteria with peptidoglycan and a membrane. Bacteria are constantly going through osmosis, or the transfer of water in and out of the cell. The peptidoglycan layer allows for structural support against this pressure. In the cell, there is a penicillin binding protein. Penicillin antibiotics find the penicillin binding protein's active site and bind to it. The beta-lactam ring opens and binds to the active site. This prevents the proper formation of the peptidoglycan layer, and the bacteria bursts. Antibiotic resistance. One form of antibiotic resistance would be when a certain bacteria contains a special protein whose active site does not bind to certain antibiotics. One example of this form of antibiotic resistance would be MRSA. The protein is penicillin binding protein 2A, and this is the beta-lactam ring. Because of the altered active site in penicillin binding protein 2A, the beta-lactam ring is unable to bind. Another form of antibiotic resistance would be horizontal plasmid transfer, or through generations. This is horizontal plasmid transfer, where plasmids are transferred from one colony to another. Plasmids contain gene information that are crucial to antibiotic resistance. As shown here, plasmids are also able to pass through generations. Overall, antibiotic resistance is a huge concern in the medical field recently. If antibiotics are continually used to treat diseases, then it's possible that bacteria are able to find new ways to resist our medication.
Medical intervention. Over the years, a bacterial infection called MRSA has slowly become more resistant to antibiotics. One of the first signs of defense to treating this condition is called vancomycin. A deadly staph infection called MRSA became resistant to older antibiotics, creating a strain of infection that is resistant to vancomycin, a new medication. The goal of our oral medication is to stop the gene associated with MRSA from producing. By stopping the production process early in the VAN-A gene cluster production, we can stop the resistant gene from reproducing. The process begins by using the enzyme that contains a complementary RNA strand to the VAN-A gene cluster to locate the correct bacterial cell. By using a complementary strand, we can locate the bad bacterial cell instead of killing good cells. The next part of the process involves using a CRISPR enzyme to inhibit translation by capping off ribosomes early in the upstream process. By capping off the ribosomes early in the process, we can stop the gene from producing in the first place. By stopping the production process, we also stop beta-lactamase, so it cannot resist antibiotics any longer. The absence of beta-lactamase now allows us to include beta-lactam in vancomycin, so we can hinder cell wall formation in bad bacteria staph infection cells. By using CRISPR, we can cut up the VAN-A gene cluster. By sandwiching the complementary RNA locator strand to CRISPR, we can ensure that they travel as a unit throughout the cell. By colorimetrically tagging the enzyme CRISPR complex, it will be visible under a microscope as a brighter color. This way, it is easier to research our findings. Our medication is intended to use before vancomycin. Our medication stops resistant gene from forming, so the use of antibiotics will become useful again.